Hello everyone and welcome to May 2016 Webinar Wednesday. As you know, this is a recorded webinar, not a live broadcast like we usually do. Sorry about that, just scheduling conflicts rule this month. I want to welcome you all, and I want to welcome our guest presenter, Dr. Brad Yoder. He is a professor of cell biology at the University of Alabama at Birmingham, and he is here today to talk with you about the results of his recently completed PKD Foundation-funded study called In Vivo Analysis of Cilia Function in the Kidney. What we're going to do today is let Dr. Yoder speak a little bit about his topic, and then I have a few questions to ask, and then we'll wrap it up. Please know that the PKD Foundation does not offer medical advice, and the information shared this afternoon is not intended to be a substitute for consultations with your health professional. Care and treatment decisions related to your health must be made in consultation with your health professional team. And with that, I will hand it over to Dr. Yoder.
All right, thank you very much for that kind introduction. My name is Brad Yoder. I've been a professor here at University of Alabama at Birmingham for about 20 years, where we have focused on cystic kidney disorders. And today I would take you through some of the results that we've obtained through the PKD Foundation support, focused on trying to understand the function of cilia in the kidney and their relation to cyst development. And my group has been working on cilia-related issues for many years now, and as many of you know, the genes, proteins involved in cystic kidney disease, such as PKD, polycystin 1, polycystin 2, and fibrocystin, all localized to the cilia, but it's not just PKD that you see this in. You have cysts that are present in a number of different human syndromes, such as Mickle-Gruber syndrome, nephronephrosis, and the interesting thing is that all of these proteins localize and function in the cilium. And so my group has been trying to understand for a while now what is the actual function of the cilium. And just to bring everybody up to speed on what the cilia are, this is an EM picture, electron micrograph picture of a tubule. And here we have what is called the primary cilium that extends off a subset of cells within this tubule. And the function of these structures, these small little microtubule-based structures, is largely unknown. And there's been several models that have been proposed. One of the main ones has been that cilia somehow play a role in proliferation, in the direction that these cells will divide in, as a mechanosensor. So you can imagine a fluid going through, urine going through the tubule here. The cilia would sense something about that fluid movement or composition of that fluid and send a signal. And the other one is some aspect of cell-to-cell communication. So these structures may be involved in sensing what this cell is doing here or here. And so I'll go briefly through this, these possible roles for the cilia in the next few slides. The connection between the cilium and proliferation has been hypothesized for a while, and the cilium, the little green structure right here, is only found on cells that are not dividing. And so for a cell to divide, what has to happen is this structure has to be reabsorbed. And there's two little things at the bottom called the centrioles. And these are needed to build a cilia, but they're also needed to orient how the cell's chromosomes are going to divide. So at the tip of these little green structures here, so these are actually the chromosomes here in blue, and these are microtubule structures that connect to the centrioles, and they are very important in separating the chromosomes as cells divide. So as long as you have a cilium on this cell, it is not able to set up this structure to separate the chromosomes in division and to make two cells. So one of the ideas has been the cilia kind of locks the cell in an anti-proliferative state. Another role that was proposed by Marco Pataglio's group several years ago was that cilia are important in dictating on how a cell divides. In a normal tubule, the cell would divide along the axis of the tubule, as shown here. And that would result in the tubule getting longer. However, when a division happens in the wrong direction, as it's shown here, the tubules get wider and a cyst forms. So this was one of the proposed models as well. And actually when he went in and looked, Marco Pataglio's group went and looked at the oriented cell divisions that happen in a cystic model versus a normal tubule. The normal tubules go this direction, the divisions do, and in a cystic model it's a random event where many of them go in the wrong direction and result in expansion of the tubule. Another model that has kind of been in favor for a while was put out by Jing Zhao's group and Suri Nali, as well as Ken Spring's group, and they proposed that the cells with these cilia sense flow, and that's shown here. And when they initiated flow, this red dye, this red color here, indicates that a calcium signal has occurred. And so the idea is that the cilia would be deflected by this flow, and that this would result in a calcium signal that the cell would sense, and some downstream activity would happen as a result of that. And we don't quite know what that downstream activity may be. But what was intriguing is if they did this in the context of a cilia mutant or in a cell that lacks the polycystin proteins, that calcium signal never occurs, and that's kind of quantified here. Normal, you get this flow-induced increase in calcium. In the context of a cilia mutant or a polycystin mutant, that never occurs. This model has recently been brought into question by Dr. Corey and Clapham's group at Harvard. So the role of cilia in mechanosensing is still a major question. And for us, what we wanted to know is, does this actually happen, any of these things actually happen inside a normal kidney? So as you notice, much of this stuff is all done in cells grown on a flat dish, where the conditions are quite different than what you would see inside a normal kidney. And so with support of the PKD Foundation, we had set out with two major goals. The first was to analyze whether cilia-mediated signaling events occur in vivo in a normal kidney, in a cilia mutant, and a polycystin mutant renal epithelium. We wanted to know if that cilia was there to sense a mechanosensory signal due to flow, and then the second goal was to see if this would alter the proteins and the genes that are present in those cells when cilia are responding to a flow signal. The issue that we were having was that to visualize cilia inside a kidney is particularly complex, and we'll get to that in a second. So we want to know what cilia are doing under normal conditions and under disease states. What signals are they sensing or signaling, sending out from these cells? And are the data that you find in cell culture-based systems or in isolated tubules relevant to what's actually happening inside a live kidney? And so, as I was saying, the cilia, trying to image cilia or to analyze cilia in vivo is really problematic. They're very small structures, and you can't see them with just a general microscope unless you're able to make them fluoresce in some way. And so what we were able to do was to generate what we call the Cellular Gfp mouse. So GFP is this green fluorescent protein, and we hooked that up to a protein that normally is targeted or resides in the cilia. And we made this so that we can induce expression in any cell that we want to, and that will turn it on, and that's what we're seeing here in this image on the right. Further, what we had to do is we had to be able to access the kidney and repeatedly access the kidney. So what we have done is to engineer a small chamber. So what we're seeing here is a kidney right there. That's a glass cover slip on the surface of this thing. And so we can put this mouse on a microscope, keep it warm and healthy and anesthetized, and we can put the objective up next to that glass, and we can actually image what's going inside that kidney repeatedly, say as cysts were developing or under changing physiological conditions. And so we have started to do that, and the first thing that we noticed, so this is looking through one of those windows, is that the cilia change with age. So this is a juvenile mouse, and these are at the same magnification. And what you'll see here is these cilia are very long in a juvenile state, but in adult states they reduce their size dramatically. And if you think of a mechanosensory model, this is actually interesting because these very long cilia respond very differently to flow or possibly a chemical or whatever is going through the fluid, the tubule here, than what you would see in an adult. The other thing I want to point out, remember this mouse is alive when we're doing this, is these cilia appear to be oriented in one direction. And I'll come back to that here in this next slide. And so this is actually a movie. I hope that these are playing, at least they are on my screen. And what you'll notice is that the flow would be going this direction. And in each case, the cilia are oriented in the direction of that flow. Now, if you think about a mechanosensor model, these guys are persistently bent under these conditions. And so what we wanted to do was to start going in and actually manipulating this and seeing if this changes the signaling activities within that cell. And first thing that we noted when we start changing blood pressure or the amount of blood flow that goes into the kidney, now you're seeing a very different response here if you just look at this guy right here, these cilia. So here would be a tubule, here would be a tubule. And the flow would be coming in this direction, right, and in this direction. And so what is happening is the cilia are sensing this flow and are bending back and forth in a kind of a mechanosensory model here. So we believe the cilia sense this change in cardiac output. We'll get back to that in a second. And the flow rate that is occurring within these tubules. So we wanted to go in and manipulate that a little bit further, and so what we did is we had these cilia, as you noticed, they were flopping back and forth. And then we added a vasodilator. Now, dopamine does, for the renal physiologist in the group, does many, many different things. But one of the things is a renal vasodilator. It will increase the renal blood flow, the amount of fluid going into the tubule, so glomerular filtration rate. It changes sodium absorption, and basically what it does is increases urine output. So it increased the amount of flow going through that tubule. And so when this movie starts again here, you'll notice that the cilia start flopping back and forth here. We flash, we're going to hit them with dopamine. And what you're going to notice is now the cilia, we increase flow, now they respond by complete deflection. However, a weird thing happened is that they actually reverse direction as well. We're still trying to figure out how that can happen, because that would argue that urine can actually go in the retrograde direction, reverse direction, which is not really supposed to happen. But we're trying to identify that a little bit further. We believe this is related to the amount of output. So here we're looking at a series of tubules, and basically what we did at this point was gave a lethal dose of anesthesia. And what you'll notice in the beginning of this movie, the cilia were actually moving back and forth, and upon here they're starting to move back and forth. Now we give them a lethal dose, and they stop. A couple little beats there, and then basically they extend out into the lumen of this particular tubule. So we believe the cilia sense changes in systemic physiology. So you change blood pressure, you change heart rate. You're going to alter how these cilia respond, and hopefully we'll be able to identify what signal that is that then changes the functions within the kidney. And we would believe that whatever these functions are going to be, that when you take away the polycystins, that will give you the same type of effect that you would have when you lose cilia as well. Oops. So we started to look at that, and my movie's not playing here, so let's try it again. All right, so in the control conditions, we note we had this cilia flopping back and forth. And we'll see if this movie's going to play or not. But here would be a big cyst, and here's another one. And what we noticed in the PKD mutants is instead of being deflected in any type of orientation or beating back and forth, within these cysts, that does not happen. For some reason, this one's not playing. We'll hopefully get it started here in a second. But here we have what is a normal tubule next to a cyst. And what you'll see is the cilia and the cysts basically stick out. This one started now. Stick out into the lumen as opposed to going back and forth like you see in either control or a normal tubule, arguing that something has changed within the flow that occurs within this cystic structure. And to begin looking at that, what we've been able to do is to inject a fluorescent dye into the tail vein. This gets filtered through the tubule, and that's what you're seeing flying through these tubules. So we can start to actually measure flow in the tubule in relation to what's going to happen to the cilia as this flow changes. If we do this in a series of still images at 10 to 45 minutes, what you'll notice in the cystic models, for example, here is a cyst over time. The normal tubules fill up with the fluorescent dye. The cystic ones do not, or they do it very, very, very slowly. And so that argues that flow within this tubule that is becoming cystic has been disrupted. And so we're going to go back and try to figure out what signals change within those cells as a result of that disrupted flow. Okay. So one of the things we've been trying to do is to understand whether the cilia play a role in calcium signaling, so the cilia are polycystins, both. And so what we are using here is what's called a biosensor, a calcium biosensor. It's a fluorescent protein that we express in these cells. Under normal conditions, it would have a very low fluorescence intensity, such as you would see in these cells, the dull cells here. Once it binds to calcium, the fluorescent intensity greatly increases. And so what you're seeing in these tubules is an increase in calcium anytime, or an increase in signal anytime the calcium signal occurs. For example, in these cells that are going right through here. And so what we're trying to do now is to coordinate these calcium fluctuations with changes in flow that may be going through here, changes in the cilia response, so are they deflected or are they oscillating, or with the presence or absence of the polycystins enduring cyst formation. And so what you can see is there's clearly a lot of calcium signaling going on. The question is, what is it doing after the calcium signal occurs? And is the cilia or the polycystins or flow a critical component of regulating that response? And we do not know that yet. We're still actively pursuing it. So then the second goal was to understand whether when you change these mechanosensory activities of the cilia, does it influence what genes turn on and what proteins are present? And so we wanted to know when you change this mechanosensory response, either by changing the flow rate, taking away the cilia, or altering the polycystin proteins, how does that impact what genes and proteins are going to be made, and how does that affect the physiology of the tubule? So basically what we want to do is to compare the genes that are being expressed when cilia are doing this versus, this one is going to take a second to play here, when they are completely deflected in the orientation of the flow. Now the issue is that it's difficult to get these specific cells relative to the many other cells that are in the interstitium between the tubules, such as fibroblasts and macrophages and other cells that are there. And so what we're started to use is this mouse that's called the TU tag mouse. This mouse, what it does is we're able to express a gene called UPRT in a specific cell based on what's called a Cree deletion. Remove this and the gene is induced to be expressed, and gene becomes expressed. And we have a Cree that is only active, this enzyme is only active in a particular cell type, in this case the collecting duct cell or the proximal tubular cell. That activates this enzyme. What this enzyme does is it takes a compound called 4-thiouracil and incorporates that into new RNA that's being made. So a gene is made into RNA, and that RNA is then made into a protein. But this compound will be incorporated into only new RNA. So here's an example of a cell in blue that does not have that enzyme, and one that's in red that does. So these little green dots here would be the incorporation of thiouracil into the new RNA. Importantly, we can now isolate this based on this little tag that we've incorporated from all the other RNA and all the other cells, and analyze what messenger RNAs are being, what genes, RNAs are being made or activated when cells are doing this our cilia are doing this versus doing this, as well as in the cyst or in the absence of the polycystins. So we tested this model and was working pretty well in some mice. So this green shows that where those cells have been, the enzyme has actually been activated. However, we noticed that in many of the cases, it was really not as efficient as we wanted. So this was one of the major technical hurdles that we've faced with this second goal of the project. As you can see, here's one tubule. Here's another one where it's all been activated. Here's another tubule where 2 out of the 5 or 6 cells in cross-section are activated, 2 out of the 5 or so. So it wasn't really quite as efficient as we were hoping it would be. And you can see that here as well when we tag the actual enzyme here with an antibody so we can see which cells are expressing it. The collecting ducts here are all red. And the non-induced cells, the ones that were not activated, are in green. And so what you can see is there's really what we refer to a chimeric effect where only one of these cells in the tubule have it, one of these. And that really makes it tough to isolate enough RNA to actually analyze what's going on. So unfortunately, with regards to goal 2, we've had to kind of re-strategize or to come up with a second alternative. Those studies are still actively going on right now. I think this has important implications in trying to understand what the cilia are actually doing under both of those conditions. So we are still actively pursuing that particular aim. So that kind of gives you a summary of what we were able to accomplish over the last 2 years of funding for this project. I think we are on the verge of understanding important new things about what the cilia are doing under different conditions, and the funding that was provided from the PKD Research Foundation was really instrumental in allowing us to do that. Based on the results that we got from the support from the foundation, we were able to submit a grant to the National Institutes of Health, which will be reviewed on June 8th. So hopefully, based on the success that we've had from the foundation support, we will be able to extend that into further funding to pursue these goals. So that is all that I have. I want to thank you for your attention during this webinar, and I'll turn it back to Alexis.
Thank you so much, Dr. Yoder. That was great. And the videos, other than the one that didn't work, they're so wonderful to just help demonstrate exactly what you're talking about. So thank you for taking the time to do that. Okay, I have a few questions that David came up with. He does apologize to everyone for not being here on this webinar. He just had some scheduling conflicts, but I have a few questions from him, and then I have a few myself more from an overall, all of your research combined questions. So let's get started with David's. The first thing, and I think something that everyone will want to know is, this work is great, it's very important, but can you talk a little bit about what it could ultimately do? So what's your ultimate goal? Will it help screen new therapies? Will it help discover new mechanisms? What's the big picture on this?
Yeah, that's a very good question. So I think one of the fundamental things, so I'm a basic scientist by training, and it's where I've spent my last 20 years. But to really understand or to solve a problem, you really need to know what the mechanism that has been disrupted. And so what I'm hoping is the ultimate outcome of what we're trying to do is that we will be able to assign a signaling pathway, a mechanism by which loss of cilia function results in the initiation and expansion of these cysts. And therefore, once we understand that, we will be able to go back and hopefully find targetable drugs or proteins that will allow us to go in and fix or reverse or even delay cyst progression. But before we can do that, we really need to understand a pathway that happens in vivo that is disrupted when the cilia are not functioning normally. So that's where I'm hoping this research will ultimately go, and We've identified a couple potential targets, and we'll see where they lead to. It's a challenge when you try to go inside a living animal to understand things, but I think that's the direction we clearly have to go.
That's great, and I think you've actually answered the second question as well, which is, will it identify new pathways and new targets for preventing cyst formation? And I think you've just answered that very well.
Yeah, I mean, that's the ultimate goal here. But first, we've got to understand exactly what the cilia are doing under the context of normal biology, and then when you take away the cilia and when you take away the polycystins, how are those pathways changed? Can we find a way of reversing those pathways?
Great. And then one final one from David. Can you talk to us a little bit about how these discoveries and how your research will lead to new research and new understanding? So it's kind of a progression, right? You make one discovery and that leads to the next and the next. So what's the outlook for this study and this research, and how do you think it's going to lead to your next discovery?
Yeah, another very good question. So from what I found is that when we finally do answer a question, there's always 2 or 3 new ones that pop up. And the big thing, I think the big change that we're doing that other groups kind of haven't done yet is we're trying to take this back into a living organism and try to understand what it's doing under normal physiological conditions. We really still have a poor understanding of what the cilia are actually doing inside the kidney. And so, our hopes are that when we identify those pathways, that it's just going to open up numerous different things to follow up on. One of the things, for example, when we hit these, we injected dopamine into these mice, we noticed that the cilia actually reversed orientation, which is not something that we ever thought we would be observing. The other thing is, the cilia oscillating back and forth. Is that related to blood pressure, or is that related to cardiac output? So I think once we understand how the cilia sense these changes in systemic physiology, I think it's going to be a big advance that will allow us to follow up on many, many different questions of what the cilia are actually doing.
Great. And then I want to just say, while you were talking, just for a moment, I clicked through some of our files and realized that I think your first grant from us came in 1998. Does that sound about right?
That sounds right.
Okay. And I think
I have been the recipient, a generous recipient of many, many PKD foundations and it is awards, and it has been instrumental in allowing me to pursue the research direction and trying to understand the connection between cilia and cystic kidney disease. And it was, with that funding that we started to make the connection between cilia and cystic kidney disease. So I'm very grateful for funding from the PKD Foundation.
And we are so grateful for the work that you do. And it seems like in '98 you started looking at novel proteins and have moved along. And I just wondered, and I know that this is on the spot, I didn't prepare you for this at all, so I apologize, but I'm just wondering if you can articulate a little bit just the funding that the foundation has provided, and has it allowed you to make key discoveries? And now you said that you've applied for funding from the NIH, and I know that you are a core center, and so that's wonderful as well. So could you just talk a little bit about that?
Sure. So the big things that have come from the foundation's support was our understanding that cilia dysfunction is a key component for many different forms of cystic kidney disease, not just PKD, but Meckel's and nephronophthisis. So with support from the foundation, we've been trying to understand the whole connection between cilia and cyst formation. And so with that, we have been able to identify new components of something called the transition zone, which is a little structure found at the base of the cilia. And for example, that is a key structure that's made of the nephronophthisis proteins and the Meckel-Gruber proteins, the MPS proteins. And that regulates what comes in and out of the cilia. And when you disrupt that structure, things like the polycystins either aren't retained in the cilia, or things that are not supposed to be in the cilia just kind of float into the cilia. And so, you know, with that, the funding from the foundation, we have been able to understand many novel aspects of cilia biology, and I think that these are all tied together. And that's one of the reasons when you make mutations in MKS proteins or mutations in PKD proteins or NPH proteins, that you get this common output, which is massive cysts in the kidney. And so I think the funding support from the PKD Foundation has allowed us to pursue a lot of novel ideas, and most of these have actually resulted in additional funding from the National Institutes of Health, where we can generate new animal models, and it was, you know, helped develop into the PKD Center that we have at UAB, in which we develop, you know, many important resources and distribute throughout the world to follow up on cystic kidney disorders. So the funding from the foundation has been really instrumental in allowing us to develop these novel resources, and to basically have a major impact, not just within my own research, but throughout the PKD research community.
All right, thank you so much. That's perfect. I think then we will call this one good. If anyone out there has questions about anything Dr. Yoder has presented today, you can email it to us at education@pkdcure.org, and we will work with Dr. Yoder to answer those questions. Otherwise, thank you all for sticking around, for listening, and have a great day.
Thank you.